Hello students, welcome to my YouTube channel. So today our topic is multiple alleles. Before we start this topic, first of all we should understand what is a gene. Gene is a segment of DNA which codes for polypeptide. This is the chromosome. This is the area where the gene is present. So this is the gene which codes for polypeptide, and the position of this gene on the chromosome is known as locus. So this is the locus of a gene. So locus is what? Locus is the position of a gene on chromosomes. And what is gene? Gene is a segment of DNA which codes for polypeptide. Now what is alleles? Alternate form of a gene are known as alleles. This is one chromosome and this is your homologous pair. So the allele form will be here at the same loci. Focus please, this is the allele form. It will be small a or it can be capital A. So alleles can be dissimilar or can be same. If suppose the individual has got same allele in its chromosome, here capital A and here also capital A, then we will say individual is homozygous. But if the individual possess two different alleles, then we say it is heterozygous. So alleles are nothing, they are alternate form of a gene. Now the question comes in the mind, ki how come the multiple alleles are there then? From the diagram it is observed ki genes can have two alleles only, but multiple alleles are there, and these multiple alleles arise because of the mutation. The mutation is nothing, change in the sequence of the bases, that is known as mutation. Multiple alleles occurs only in population. Multiple alleles means when more than two alleles of a gene which controls a particular trait occupy the same locus. Locus means same position. They occupy the same position on a chromosomes, on a homologous chromosome. We call all these alleles as multiple allele. Is it clear? We call all these alleles as multiple alleles. What is the reason behind the occurrence of multiple alleles? Multiple alleles arise due to mutation. This I discussed it also. The reason for the multiple alleles are mutation. Now, if you have to study the multiple alleles in human, the best example of multiple allele in human is ABO blood group inheritance. ABO blood group inheritance is an example of multiple alleles in human. Now we will start with the ABO blood group understanding. Now see, first of all, the scientist Carl Lenster, he discovered two kinds of antigens. Antigens are A and antigen B. They were present on the surface of RBC. Means he discovered that on the surface of RBC, one A antigen is there and surface of RBC has got B antigen. Antigens are nothing, they are glycoproteins. Now based on the presence or absence, there are three kinds of blood groups are there. Blood group A, blood group B and O blood group. So all these blood groups are depending upon the presence of an antigens. Type A blood group means it will have antigen A. Type B blood group means it will have antigen B. Type O blood group means it will not have any antigen. Now the fourth blood group, which we say AB blood group, that fourth blood group was discovered in 1902 by the two students of Landsteiner only, that is Van de Kessel and Sturley. They discovered the fourth blood group. So which is the fourth blood group which was discovered? That was blood group AB. This was the fourth blood group which was discovered, which is also known as universal recipient. O is known as universal donor. What is the biochemistry behind these blood groups? How these blood groups are coming? Bernstein in 1925, he discovered that inheritance of blood group in human is by a number of multiple allele series. So blood group is because of the multiple alleles. Now alleles, if you see, the genes are located on the chromosome 9th. For blood group, genes are located on the chromosome 9th. And the gene is actually, we are representing it by symbol I. The gene I exists in three forms: IA, IB or IO. So your gene I is existing in three forms: IA, IB and IO. So here blood group is controlled by three alleles. Blood group is controlled by three alleles. So blood group is a case of multiple allele. Now see, what is the biochemistry of the blood group? See the genes. This is a gene IA. This gene will produce this enzyme, that is N-acetyl galactose transferase enzyme. So gene will produce N-acetyl galactose transferase enzyme, and this enzyme will add N-acetyl galactosamine to H antigen to make the blood group A. It will add H antigen to make the blood group A. This is a simple, I'll explain it this way. This is the blood RBC, which is having, let us assume, this is the H antigen present on the surface of RBC. This is H antigen. This is your H antigen. Now to this H antigen, if suppose we are adding to this H antigen, when we add galactosamine, when we add N-acetyl galactosamine, then the blood group, this is your H antigen, and now I am adding, this is your N-acetyl galactosamine, that is N-acetyl galactosamine. Then this becomes your A antigen. It becomes A antigen and blood group A is made. Now gene IB. Gene IB will add galactose transferase. It will make galactose transferase. This is an enzyme which will add galactose to H antigen. Again see here, this is your RBC and this is your H antigen. Now, if suppose we are adding galactose. When we add galactose, this is a galactose. When we add galactose to H antigen, then it becomes blood group B. B blood group is made. So here we have focused it कि B blood group is made. So H antigen is always present on the RBC. H antigen is always present on RBC. Now what we add? If we add N-acetyl galactosamine, it becomes blood group A. If we add galactose, then it becomes blood group B. From this it is clear that both the alleles IA and IB are able to produce their sugar. Means they are able to produce an enzyme, and enzyme is able to add the sugar part to the H antigen. It means from it is here it is clear that both the alleles are expressing. Both the alleles are expressing. In the theory, if you see, I have given it this way here. This is a theory. You can note it down. So from this, from the phenotype combinations, if we see blood group 4 phenotypes, from the phenotype combination, it is clearly seen that alleles IA and IB are dominant to IO and they are co-dominant to each other. Both are dominant to each other. So best way to represent, we can represent the relationship in all the 3 alleles in this method. This method indicates that IA and IB both are dominant and they both are dominant over IO. So from this, if you see this relationship, It indicates blood group is a case of codominant also, and it is following the Mendelian dominant recessive relationship. Mendelian dominant recessive relationship. Different types of blood groups. If we I've made this map. So different types of blood groups are there. If we find total 4 phenotypes are there. Type A phenotype, type B, and type AB and type O phenotype. But genotypes are total 6 in numbers. Genotypes are total 6 in numbers. First genotype, second, third, fourth, fifth, and sixth genotype. So blood group A can have 2 genotypes. This blood group can have this genotype as well as this genotype. Same blood group B can have again 2 genotypes. But AB can have only 1 genotype and O blood group can have only 1 genotype. So now if you see, the blood groups are depending upon the presence of antigens. We have already discussed antigens are nothing sugar. So blood group A means it is making A antigen. Blood group B means it is making B antigen on the surface of RBC. On the surface of RBC. AB blood group means both antigens are present. O blood group means neither A antigen nor B antigens are present. Antibodies are always present in the plasma and they are opposite. Antibodies are always present in the plasma and they are always opposite to the antigen. If suppose antigen A is there, if the blood group has got antigen A, then antibodies will be B. If blood group is B, antibodies will be AB. If blood group has got both antigen, no antibodies will be there. That is why type AB blood group, we say it is universal recipient because it does not have any antibodies. And type O blood group, it has got both antibodies but it does not have any antigen. Because of this, it is known as universal donor. So if you see ABO blood group, it is having 4 phenotypes and 6 genotypes. So here I have mentioned the formulas for the phenotype is n plus 1. Means whatever number of alleles is given plus 1, if you add, you will get phenotypes. For example, in case of blood group, if you see the number of alleles were 3. So phenotype formula was coming. Phenotype was coming 3 plus 1, 4 phenotypes, which we have already seen. Yes, we are getting 4 phenotypes in case of the blood groups. Now, if you see the genotype, genotype is n by 2 into n plus 1. So if you substitute the values, 3 by 2 into 3 plus 1. So it will be coming 3 by 2 into 4. So you will get the answer as total 6 genotype. So blood group is having 6 genotype. Now another examples with respect to the multiple alleles. Now note down other examples of multiple alleles. Eye color in Drosophila. Drosophila has 15 alleles. Next example is coat color in rabbit, which is having 4 alleles. So these are the examples of the multiple alleles apart from the human blood group. So I hope this will help you to learn for your NEET exams. Thanks for watching. Please subscribe the channel and like it.